Hi everyone. Today we'll start to look at the inheritance topic from AQA topic 7. So in this section we'll be covering dominant and recessive alleles, codominance, multiple alleles, sex linkage, and how to interpret pedigrees. All of these concepts will first be looked at in a monohybrid cross where we're only looking at the interaction at one locus. But all of these concepts can be applied to dihybrid crosses too, and this is what we'll go on to look at in part 2. So first of all, we need to look at some definitions. The first one being genotype. This is the genetic constitution of an organism. What we mean by that is the alleles that a particular individual carries for a particular gene. The genotype then influences the phenotype, and this is the expression of that genetic constitution. But the phenotype can also be influenced by the environment. It's really important that we get our terminology right. Lots of students will lose marks because they use the term gene instead of allele. So we need to be specific. Alleles are different forms of the same gene. The best way to explain this, like with all the genetics, is with an example. So we'll look at fruit flies, which are a model organism used in genetics. So we're looking at the wing shape gene. So wild type wings are coded for by big N. So the allele is big N. Wild type wings are the standard wings that are produced on a fruit fly. The lowercase n, little m, codes for vestigial wings. These flies develop stumpy wings and can't fly very well. So here, big N is dominant, which means if it's present, it will always be expressed. Little n is recessive. This means it's only expressed in the absence of big N. Be really careful where you're defining recessive, and we don't say that 2 copies are needed. That might be true for a diploid species, but the best way to explain it is say it's only expressed in the absence of the dominant allele. So what this means for our phenotypes is the homozygous dominant, where there are 2 big N's, one inherited from each parent, will have wild type wings. Heterozygote will inherit a big N from one parent and a small n from the second parent. That will also have wild type wings because one big N is present and being dominant, it is always expressed. The homozygous recessive individuals carry 2 little n's. So here we have an absence of big N, and this means the recessive phenotype is expressed. So these flies will have the vestigial wings. Let's look at the inheritance of wing type to model a monohybrid cross. So this is where we're looking at the inheritance just at one locus, so just one gene, and in this case we've got a dominant allele and a recessive allele. So first of all, we look at the parents and we can see we've got both wild type wings. So we're going to state the phenotype and also the genotype of the parents. In this case, they're both heterozygotes, so they carry a dominant big N and a recessive little n. And I've colour-coded these alleles so we can track them through the cross. So our next step is to show which alleles are in the gametes. So in the first parent, half of the gametes will carry the big N, so the one that's teal coloured. The other half of the gametes will carry the lowercase purple n, the little m. In the second parent, half of the gametes will carry the blue big N, and the other half will carry the orange little m. And then we're going to draw out a Punnett square. So this helps us to show the different combinations. I'd really advise you draw these out, even if you think you don't need to. It just means that you're not going to miss any combinations. So we're going to list, down the side, we list the gametes, the alleles from the gametes from the first parent. So we've got our big teal N and we've got our little purple N. Across the top, we're going to list the gametes from the second parent. So we've got the big blue N and we've got the little orange N. And then we show the different combinations that could be combined at fertilization. So we could end up with big N, which would give us wild type wings. Big N, little n, which would give us wild type wings. Another big N, little n, which gives wild type wings. And then we've got this 1 in 4 where we've got little n, where the fly would have vestigial wings. So what we can see here is if we start with 2 heterozygous wild type parents, so they would both have the wild type wings, but they both carry the recessive allele, there's a 1 in 4 probability of producing offspring with the recessive trait, so with the vestigial wings. The concept of codominance is where there is no recessive and no dominant allele, and in fact both alleles are expressed. So within any heterozygotes, we see a combination of both phenotypes being expressed. Let's look at an example. So here we have camellias. So camellias, in the parents, we've got red flowered plants and we've got white flowered plants. So the parent with the red flowers will have a genotype of big R, big R. The white flowered plants will have a genotype of big W, big W. And as you can see here, we're using a different letter to represent the different alleles. Within the gametes, as they're both homozygous, there is only one type of gamete for this gene. So the red flowering plants will produce gametes that carry the R allele, while the white flowering plants produce gametes carrying the W allele. So what happens at fertilization and what do the offspring look like? Well, their genotype will be RW, so they are heterozygous, but here their phenotype is not the same as either of the parents. And what we see is that both alleles are expressed, and we can see a combination of both phenotypes. So the flowers will have part red and part white colour, so we see like a kind of patchy effect. Now we're going to cross these offspring. So we're going to start with 2 parents that are both heterozygous, so they both have red and white colouring, and their genotypes are both RW. This means that each parent will produce 2 types of gamete, so parent 1 will have half the gametes carrying the R allele, while the other half carry the W allele. Parent 2 will have half the gametes carrying the R allele and the other half carrying the W allele. And then we produce a Punnett square to show our offspring. So we're going to get 1 in 4 with RR, so these will be red in colour, 2 in 4, half with RW, so with the same phenotype as the parents being both red and white, both phenotypes being expressed, and then we'll get 1 in 4 with WW having white flowers. So here we're getting a 1 to 2 to 1 ratio. Our next concept is that of multiple alleles. So far we've looked at examples that have 2 alleles at a particular locus, so 2 alleles for any one gene, but in some cases some genes can have more than 2 alleles. So each individual will still only carry 2 alleles, and so we've got different combinations. So the example that's often used for this is ABO blood types. So the ABO blood group is determined by the antigens that are present on the surface of red blood cells. So for blood group A, antigen A is expressed, so that would be the phenotype. The A antigen is expressed on the surface of the red blood cells. In blood group B, only the B antigen is expressed. And what we can see in the next group of AB, both are expressed. So this, like in our last example, is codominance. The fourth group here is group O, where neither are expressed. So in terms of how the alleles interact, we represent them as IA, IB, and IO. So in this case, we've got 3 alleles. IA and IB are codominant. So where we've got blood group AB, both are expressed. However, both are dominant to IO. So let's see how this would work in terms of genotype to phenotype. So if someone inherits IA, they're of course going to be blood group A. If they inherit IO, because IA is dominant to IO, they will also be blood group A. Someone inheriting IB would be blood group B, expressing the B antigen on the surface of the red blood cells. If they inherit IB with an IO allele, because IB is dominant to IO, they would also be blood group B. Blood group AB can only be brought about if the individual carries both IA and IB. And then blood group O, being recessive, the only way blood group O can be expressed is in the absence of IA and also in the absence of IB. So these individuals must be IO. Let's look at a cross to exemplify this. So first of all, our parents, we're going to cross an individual of blood group A with an individual of blood group B, and both of them are heterozygous. So the genotype for the individual with blood group A is IA, IO, so they're also carrying that recessive allele. The individual with blood group B are carrying the dominant IB with the recessive IO. So their gametes. Individual 1 will have half the gametes carrying IA and half the gametes carrying IO. The second individual, half the gametes will carry IB, while the other half carry IO. And then we put this into a Punnett square to determine the phenotype and genotype of our offspring. So we're going to have all 4 blood groups found within the offspring. The genotype IAIB will give us blood group AB. IAIO gives us blood group A. IBIO gives us blood group B. And finally IOIO, we've there got blood group O. So what we're seeing here is all 4 blood groups found in a 1:1 to 1:1 ratio. The final concept we'll look at for our monohybrid crosses is that of sex linkage. So this is for genes that are located on the X chromosome, and for this reason the sexes are affected differently, as females carry 2 X chromosomes and so have 2 copies of each of those genes, while males only have 1 chromosome, 1 X chromosome, so they'll only have 1 copy of each of the genes on it. So let's look at an example for this, and here we're going to look at the inheritance of red-green colour blindness. So we're going to start off with the parents that both have full colour vision. For sex linkage, it's really important that we specify the sex. Although it might seem obvious, that's part of the phenotype. So within our phenotypes, we've got a full colour vision female and a full colour vision male. The genotypes, we're going to specify the X's and Y's in terms of the inheritance of sex. So for the first parent, we've got X big N, X little n. So the N's are used as superscript on the X chromosomes. In the male, we've got X big N, Y. We don't need to put an N on the Y because there is no N gene on the Y. This means that males can only carry 1 copy of that gene. So what we can see here is the female is heterozygous. The male either carries big N or little n. It can't be heterozygous. So let's look at the gametes. So in the female, half of the gametes will contain X big N, while the other half carry X little n. In the male, half of the gametes will carry X big N, and the other half will carry the Y chromosome, so no N allele. Then we do our Punnett square to see what happens at fertilization. So as we can see, we could have X big N, which gives us a full colour vision female, and we specify the sex because this is part of the phenotype. We could have X big N Y, so this will give us a full colour vision male. X big N, X little n gives a full colour vision female, and X little n Y gives us red-green colourblind male. So half of the male offspring will have red-green colour blindness. Be careful with these questions because sometimes they will ask for the probability of sex as well as the phenotype that we're looking at. These conditions are more common in males than in females because they only have one X chromosome. We'll finish off with looking at some pedigrees and the sort of patterns to look out for each of the different types of crosses. So a pedigree is like a family tree where circles represent females and squares represent males, and then we can shade in the circles and squares to track a particular phenotype or genetic condition. So here we will be looking at a condition that is autosomal dominant. So anybody that carries at least one dominant allele would have this phenotype or condition. What we're looking for here are carrier parents. So they have the condition, it being dominant. However, as both also carry the recessive allele, they could produce an unaffected child. If we compare that with autosomal recessive, we see the same pattern but in reverse. So here, because it's a recessive phenotype or condition, anyone to have the condition would need to carry 2 copies of the recessive allele in order for the dominant allele to be absent. So again, we've got carrier parents, but this time they are unaffected. However, both carry the recessive allele and so both could pass on and have an affected child. Pedigrees can also be used to interpret sex linkage. So if we have sex-linked dominant phenotype, we're looking there for daughters of affected fathers will always be affected. The reason for this is the father only has one X and he will pass that on to all of his daughters. So if he carries the dominant allele on his X chromosome, he will pass that on to all daughters. Here we can presume that the mother has no dominant allele, so she is X little b, as she is not shaded in on the pedigree. So she would pass on the X little b allele, and all the daughters would be heterozygous. For sex-linked recessive, we're looking for mothers and sons. So for here, any affected mothers, their sons would always be affected. So again, the males only carry one X chromosome. So here we can see the father is unaffected, and as this is a recessive condition, we can presume that he carries the dominant allele on his only X chromosome. The mother is affected. So with it being recessive, this tells us that she only carries recessive alleles on both X chromosomes. That means she can only pass on X little b. Any daughters can receive the X big B from the father, but the sons receive the Y chromosome with no B allele, and so all the sons in this case will have the genotype X little b Y and will be affected by the recessive condition or phenotype.